Okay,
All
so
right.
let's get started. So let me just shortly explain. My name is Rasmus Bro. I'm from University of Copenhagen, and today we have Paul Gemperline and Farnoosh Koleini, who used to both be at East Carolina University, where they just published this very interesting combination of Tucker and ASCA that I'm looking very much forward to hearing. Now, if you have questions along the way, feel very free to just interrupt because they prefer that sort of way of interacting. So feel free to interrupt also by just opening your microphone. That's all good. And with that, looking forward.
All right. So thank you so much for the invitation to give this presentation, Rasmus. I'm Paul Gemperline, and I'm pleased to talk about this project that we've been working on for about 4 years. Our aim was to develop a novel method combining ASCA and Tucker3 models to explain high-dimensional data. And Farnoosh Koleini, who is with us today, finished her master's degree at ECU last year, and this project was the subject of her master's thesis. Farnoosh, you want to say hello?
Thank you. Hi, everyone. I'm Farnoosh Koleini. Currently, I'm a PhD student at UNC Charlotte. I had a pleasure to work with Dr. Gemperline for my master's, to work on Tucker3 and tensor decompositions. And I'm so happy to be here, and thank you for joining us.
Yep. So after I retired about 1.5 years ago, and my last 14 years included a heavy administrative load. So I stayed on as an adjunct to help Farnoosh finish, and now that I'm retired, I have more time to engage in mental activities that I enjoy, which includes applications of chemometrics to multivariate and MATLAB programming to multivariate problems. So the main objective of this study is to illustrate the complementary nature of ASCA and Tucker3 analysis on designed data. By that, I mean, if you have a multivariate data set with an experimental design of at least 2 factors, then the data matrix can be folded into a multi-way tensor. ASCA can then be used with, which is a multivariate ANOVA method, can be used on the unfolded matrix, and Tucker can be used to analyze the data folded as a tensor. We call the combination of these two Tusca. So briefly, there's an outline for my talk today. I'm going to introduce Tusca with a 1992 blue crab data set. Maybe some of you I know are familiar with it. It's a small data set that I'll speak about that a little bit later. We'll introduce a Monty data set, which is a gene expression data set, which is a fusion of messenger RNA, methylation, and microRNA to create a tensor for classifying 4 different subtypes of colon cancer. We'll show how Tusca works on that. We'll talk about a novel Bootstrap analysis procedure that we've developed for aligning eigenvectors in the Tucker3 models and core values. We'll briefly discuss the effect of preprocessing options on classification. And we'll use Bootstrap confidence intervals for visualizing the data and begin maybe just a little bit of interpretation of the results to identify biomarkers, which is our ultimate aim. So as I mentioned, we developed Tusca using a small, well-understood blue crab data set that was published in 1992. Diseased crabs were being caught in the Pamlico River in the vicinity of a phosphate strip mine, which had a permit to discharge about 20 parts per million fluoride from processed waste. Greenville is up here, so we're about 1 hour drive from this phosphate strip mine. Our hypothesis was at the time that fluoride waste was solubilizing minerals present in the clay and silt of the riverbed that aren't normally soluble at river pH. So we collected, with the help of some fishermen, 16 healthy crabs, 16 diseased crabs, and 16 healthy Albemarle crabs as control. From those crabs, gill tissue, muscle tissue, and hepatopancreas tissues were taken and assayed for 28 elements by ICP atomic emission spectroscopy. And you should just know that these diseased crabs had holes in their shells, some of them almost as big as a coin. It's called burnt shell disease, and these holes were infected with chitin-eating bacteria. Chitin's the protein as part of the shell. And these were very, very sick animals. There was a happy ending to this story. In 1991, a process waste recycling stream was put online. So fluoride discharge ceased and the disease went away. So here's an illustration of the three-way data array with 48 crabs, 28 elements, and 3 tissue types. And I like this. I like to visualize these models with these kind of block representations. It helps people understand. It helps me understand this, how to visualize this. So in our 1992 paper, we calculated a model with 4 eigenvectors in G, 6 eigenvectors for H, and 2 eigenvectors for E, a 4x2 model. It explains 71% of the variance, and we selected the number of factors based on scree plots and a visual interpretation. So when we unfolded the residuals like this, we found out that there was still structure in those residuals when we analyzed them by ASCA plus analysis. When we, let's see, so then our goal then became to determine the number of factors in the Tucker3 model with ASCA plus. So our goal then is to find a Tucker3 model that on the one hand uses enough factors in each mode to explain all of the data, so a statistically sufficient model, and on the other hand is as simple as possible, a parsimonious model. So we made a grid search. We made a grid search with all of possible combinations of models with Tucker3, from 1 to 10 factors for G, 1 to 10 factors for H, 1 to 3 factors for G. And then the resulting residual matrices were unfolded and tested for significance using ASCA plus with 10,000 permutations. Computational effort for that is maybe with MATLAB Parallel Toolbox is maybe about 4 to 6 hours. The simplest model that explained all the contributions was a 373 model with the p-values shown there, respectively on factors A, B, and the interaction between factor A and B. So let's see. Why is not? There we go. So I always like to plot the data and take a look at it. So here's a plot then of the Tucker residuals for the 452 model. Red shows the gill residuals, green shows the hepatopancreas residuals, blue shows the muscle residuals for the different elements here. And you can see by the histograms that there are some offsets and that there's still some structure in the residuals. This is exactly the structure that ASCA detected, and that's what it was designed to do. So then, let's see, why am I not? There we go. So then after making the 373 Tucker3 model, then you can see that the residuals are now all centered on a mean value of 0. We did find a couple of outliers that's well described elsewhere. So we found this, we have this now useful method, and we think that I did want to say one other thing about this. When we looked at clusters in scatter plots of the elements, we observed clustering of certain metal ions like aluminum, cobalt, and manganese. These are naturally occurring in the clay, and they're normally insoluble. So we think then that they were being solubilized by the fluoride ions, as I had said before. We did find something very interesting. We saw clusters of copper and sodium. We saw that the elements copper and sodium were clustered together. And this was puzzling to me at first. So when I spoke to our biologist collaborator, he told us that he reminded us crabs can move from highly saline environments with different salinity rapidly. And in order to in order for them to do that, they have to adjust the amount of copper in their blood, the hemolymph. Their blood is copper based. They adjust the amount of copper to offset the changes in salinity, so that the osmotic pressure, the change in osmotic pressure would be equalized. Otherwise, it would cause cell membranes to rupture in vital organs. So this finding was particularly gratifying to us. When I went back then and looked at PCA scatter plots of just one tissue type at a time, and those the copper and sodium clustering was not observed. So here's the thing. We only observed this copper and sodium clustering when we analyzed all three tissue types together in a Tucker3 model. And I think it really illustrates beautifully the unique power of multi-way models, in this case, the Tucker3 model. Factors that are extracted, which are really triads of eigenvectors, explain the maximum amount of variance possible while simultaneously modeling correlation between each mode. And this is that extra information that comes to us in the form of correlation between these three modes that's not accessible in two-way methods. So understanding this unique power of Tucker3 models, we thought it might be useful for biomarker discovery in multi-omics studies. And so we sought out to identify some data sets in the literature that would be amenable to this kind of analysis. And Farnoosh is going to describe that next.
Thank you. All right, so we developed the method and we tested on the blue crab data. So now it's time to talk about the Monty project. So we got this data, the Monty data set, from a paper in 2021, which is a multi-omics non-negative tensor decomposition framework for gene-level integrative analysis. And in that data set, which is coming from the Cancer Genome Atlas program from NIH, we had specifically 315 patients, colon cancer patients, with 4 main subtypes, CMS1, 2, 3, and 4. And we were working on 3 different omics types. You see gene expression, methylation level, and mRNA expression, all of them with different number of features, but the same number of samples. For example, for the gene expression, we had specifically 14,514 number of features or variables. Next slide, please. Okay, so this is a general framework we had for this project. First, what was our aim here? We wanted to discover the gene regulatory multi-omics features, or specifically, we call them biomarkers. And for do that, we had to do first the data preparation and data preprocessing part. And we got this idea from that. Again, the Monty paper for preparing that tensor data. We had to compute it and assigned all the omics like matrix values with respect to each gene. And you may want to ask us why multi-omics type or multi-omics like view or approach versus single omics, because we just told you we developed a Tucker3 model, that this model is able to extract the maximum amount of variance per factor, just like PCA, but simultaneously modeling correlation among all 3 modes. For example, here we have omics, genes, and samples. This is kind of powerful. Okay, after creating that tensor, we implemented a Tusca, which was the combination of Tucker3 and ASCA plus. And you see in that figure, we're going to decompose that tensor through the array to a core matrix or core tensor and 3 different loading matrices: layer mode, column mode, and row mode, and then visualizing each eigenvectors of those 3 different matrices to do some visualization and interpret their results that we will talk about it in the next slides. Next slide, please. Next slide, please.
Yeah, I'm trying. There we go.
Okay. Great. So let's talk about a little what's going on in a cancer cell and what is actually in a normal cell, what it should be. So you see in that figure for the DNA methylation patterns, in the normal cell, we have like the promoter DNA methylation patterns in carcinogenesis and the promoter of intergenic and intragenic mRNAs. I mean, between genes and within a gene is unmethylated in normal cells. So the transcription is ready. But in cancer cells or when the tumor growth is happening in a cell, those intergenic and intragenic mRNAs is aberrantly methylated or hypermethylated. So it's going to block the transcription in the cell. So this is actually what's going on in a tumor, like in cancer cells when we have tumor growth, and what we don't have in a normal cell. So this is about the methylation happening in there. Next slide, please. So I told you we had 3 different matrices with different number of features, one for gene expression with 14,513 number of features or variables for genes. And also we had 2 other different data sets, the methylation level and mRNA. So we had to create a tensor with the same size number of features for those 3 different layers or matrices. So there was a data preparation part for this based on the Monty paper. So the methylation data frame converted to a gene level data set according to the gene corresponding to the promoter region. So the methylation sites per gene were average, and just creating the gene level methylation data set with the same number of features as gene expression data, which is the 14,513 number of features with the same number of samples. And again, next slide, please. We had like the same process for the next data set, which was the mRNA data set. The actual number features for that was about 2,000 number of variables or features. But again, we had to calculate, or it's better to say the mRNAs were bundled and the geometric mean calculated respect the gene. And we again created that tensor in a gene level value, which is the same number of features and number of patients again. Next slide, please. Okay. So the data was prepared, and the next step was data preprocessing part. So the QNOR or quantile normalization is a kind of widely used preprocessing method or technique. It's for removing the technological noise in microarray genomic data. And it's a kind of simple procedure. It has 4 main steps that I brought it here for you with a picture. And you see in the picture in the first one, the first step is ranking gene of each sample by magnitude, and then calculating the second step, calculating the average value for genes or agents of the same rank, and then substituting those the value of each genes of a specific rank with the average of that rank. And at the end, again, reordering the genes of each sample to the original order. So that was the general process for QN or quantile normalization preprocessing method. So the data is ready, preprocessed, and now it's time to implement Tucker3.
So once again, I like to have this visual representation of the Monty data set and the Tucker3 model. I find it useful to keep this image in mind when I show some subsequent visualizations. First of all, it's the Monty dataset is not from a balanced design. The size of these blocks for CMS 1, 2, 3, and 4 are drawn in proportion to the number of subjects or patients in those. And you can see the dimensions of the tensor with the three different layers. And then this is the model that we're going to compute. And our objective here is to determine the number of factors in the Tucker3 model with ASCA+. We want, again, a model that explains all of the variance due to the experimental design, but on the other hand, is as simple as possible. So we did a grid search for all possible combinations of Tucker3 models with 1 to 15 factors on G, up to 25 on H, up to 3 on E. We skipped models that didn't have Kruskal rank that would give a unique model, so it saved a little bit of time there. We did the unfolded residual matrix testing with ASCA Plus at 2,000 permutations. So it's a pretty large data set, about 2 gigabytes just for the data, just for the tensor by itself in double precision. We were fortunate to be able to run these models on NIH supercluster using MATLAB's parallel toolbox. The best model that we found, the simplest model, was a 9 by 19 by 3 model. And now I want to say a little bit about ASCA Plus with nested designs. We have, let's see, why did it do that? There we go. So we have several factors involved here. Factor A is the type of cancer, CMS 1, 2, 3, 4. This is unbalanced because we have different numbers of patients or subjects per category. Factor B is the omics type. We have 3 levels for that. Factor C is the subject or patients, which is nested in factor A, the cancer type. So it's a random factor that measures the intersubject variance nested in factor A. And then we have also interaction A with B. So then this is the model. You'll notice that it's written as a regression problem because it's unbalanced design. So solving for the ASCA mean vectors, we get solve the generalized linear model, and unconstrained permutations on the residuals of the reduced ANOVA models were used to estimate the significance. This next slide, let's see, next slide shows the how the sum of squares is partitioned. And these permutation tests were performed with code from Pepe Camacho. He's been a really great collaborator with us on this project. So the total sum of squares corrected for the mean shown here. It's partitioned into sum of squares for factor A, factor B, the nested factor, and then the interaction factor, and then the residuals. So for testing for significance for factor A, we use the ratio of the sum of squares for factor A divided by the sum of squares of subjects nested in A. And for interaction B, we have the sum of square. Oops, there's an error in that formula. That should be a SSB in the numerator, not SSA.
Merci. Là, je savais pas que tu étais là.
Okay. Was there a question? Okay. So we then do this bootstrap. This is a summary of how we do the bootstrap. We unfold for our Tucker model. So we have a 9 by 19 by 3 Tucker model. And we unfold the residuals of that. The unfolded residual matrix size is shown here. And we resample the residuals column-wise with replacement to form a new residual bootstrap sample. That's added to the model to produce the bootstrap sample, and then a new Tucker model, 9 by 19 by 3 model is computed. There's an ambiguity in Tucker3 models. The order of the eigenvectors can be shuffled, but the model is still, and the core values can be shuffled, but they're it's still the same model. So we used the order and the order and algebraic signs of the core values and loadings change, but the magnitude and shape stays the same. So we use correlation analysis, and for this, Stuart Hugler really helped us develop the MATLAB code that does this very nicely and efficiently. We sort the eigenvectors and the order of the Bootstrap in each of the Bootstrap iterations to match the original Tucker3 model. And then we still have a sign ambiguity in the Tucker model to deal with. Let's see, for example, if for a Tucker3 model, say with a core value, we have the product of 4 items: a core value, a vector G, a vector in H, and a vector in E. And let's say, for example, if the algebraic sign of the core of the first 2 items is minus minus then plus plus, the algebraic sign order can be changed. It could be minus plus minus plus, or minus plus plus minus. Any permutation of that would give the same overall net contribution to the model, to the sum of squares. So there's could be some really complex bookkeeping involved, but we use the concept of parity. So examine the sign of the core value and the sign of the G loading, and if the core value sign doesn't match the reference model, we just flip that sign and flip the sign of the corresponding vector in G. And by doing that, we don't have to do any complex bookkeeping. If you go through the whole model, in the end, the sign flip problem is corrected. And that's shown graphically here. This is the core element 342, with the sign flip, about 50% of the models plus, about 50% of the models minus. And after correcting for the sign flip, this is the distribution we have for that core element. So this is a big model. The 919.3 model has 513 core values. That's a lot of parameters to estimate. So we want to look at the Bootstrap confidence intervals of these core values to determine which ones we think are significant or not. So we've shown here is some of the the largest core values here and some smaller ones shown here. And the ones above in green are all significant at the 95, highly significant. There's the Bootstrap confidence interval. The ones down below in red are not significant. And it's important at this point to remind ourselves, each triad is a mutually orthogonal to all of the other triads. So the Tucker3 model there's 513 orthogonal tensors. That's pretty amazing to me by itself. The variance explained by each triad can be visualized and interpreted independently. That's important. Because they're orthogonal, we can interpret them independently. And so we focus our efforts on triads, interpreting triads with large variance and highly significant on the Bootstrap confidence interval. So here's an example of a core value that is not significant. The histogram from the Bootstrap confidence interval is shown here, 95% confidence interval in green. And you'll see the value 0 falls within the 95% confidence interval, indicating that this core value is not significant. So on to some visualization of the loading vectors. This is a plot of G2 from so this is the second eigenvector from matrix G, and shown in black is the original eigenvector. Shown in cyan is the 95% confidence interval from the Bootstrap, and shown in magenta is 2 times the 95% confidence interval. It's really important to guard against overfitting if we want to do some classification, solve some classification problems. With 14,500 genes and 513 core values, estimated parameters, the risk of overfitting is really significant. So this bootstrap procedure then really helps us to avoid that. So we can see there seems to be some differences in these different on this eigenvector, on this loading of different for the different cancer types. Here is let's see, there's this G3, the third eigenvector. Again, there appears to be some separation. And here's the fourth one. There appears to be some separation by class, but there's something interesting going on here. We see some patients with very large values and highly correlated to one another. So we want to maybe take some time and look at the demographic factors or other factors involving those patients that causes their gene expression in this multi-omics data set to be different from the other ones. Are they smokers? Do they have some other kind of illness at the time the sample was taken, the tissue sample was analyzed? We haven't taken that deep dive yet, but this is certainly something that can help us investigate that property. So then, let's see next. So we took all of those loadings, all 9 eigenvectors from G, and used them for classification by the MATLAB classification toolbox. This is pretty preliminary result where we need to be a little, I'll say a little bit more about that in a minute, but the best overall classification rate was 88% with a 5-fold cross-validation. The best method was optimized neural net. And we studied many different pretreatment strategies. The best pretreatment strategy was mean correction by omics type. Let's see, so back again, looking at this. So we just looked at some interpretations, some visualizations of the column mode eigenvectors. Next, we're going to look at row mode eigenvectors, which is going to tell us something about the genes. Oh, so sorry, that's not the genes. I'm going to look at, can I skip something? No. This is, do I have, let's see, no. Oh, okay, your genes comes later. All right, so we're going to look at the layer. This is the layer mode eigenvectors E. And because we subtracted in our pre-processing, we subtracted the mean of each omics type, we see that these layers are somewhat orthogonal to each other. The first loading of E has a very large value for microRNA. So any combination of any triad, which includes this loading, is going to be responsive to modeling microRNA. The second loading is almost all methyl. The signals for microRNA and RNA are down in below the confidence level. And for the third mode, we see a strong signal for the RNA gene expression and not very significant signals for methyl and microRNA. So that's going to help us when we try to make an interpretation of the gene mode eigenvectors. So now, let's see, we're going to make an interpretation of mode number of combinations of eigenvectors using 1 here. This is microRNA is vector number 1. So this combination of eigenvectors 1, 1, 1 models a significant signal for microRNA. And this combination of vectors 2, 8, 1 also models a significant contribution. This one is a large positive contribution to the signal. This has a negative contribution to the overall signal. And these other vectors aren't modeling microRNA. when we go back and look at eigenvector 2, we see methyl has a strong response on that. So any combination of triads, which includes that vector 2, those triads are going to be modeling the methylation signals. So here are these 3 triads. And lastly, the other one, when we're trying to interpret the genes represented by this triad, we're going to be looking at effects on gene expression. So are there genes that correlate with that are correlated? There's 14,000 of them. The confidence intervals are plotted here. And so we're just going to take a look at a snapshot right here in this region so we can see a little more clearly what's going on. So the red region is the 95% confidence interval. Yellow is 2 times the 95% confidence interval, and green is 3 times the 95% confidence interval. So if we want to avoid overfitting in the way we make an interpretation of genes in this data set, we want to focus in on genes that have a high signal compared to their confidence interval, and we want to ignore the ones that have a low signal compared to their Bootstrap confidence interval. And doing that, we have now selected some genes. So this shows then 15 of the 19 loadings on H, and they've been sorted in order of the normed gene confidence interval. So the value here in position 1 is 61 times larger than the 95% confidence interval. And we can take these sorted values. We set a threshold. We looked at the top 13 genes in each column. We set a threshold of 6 times the 95% confidence interval for selection of genes for further investigation. And we can do a table lookup. So these are the indices. We sorted those arrays. So this is the indices of the genes that we want to begin to look at. And here they are. We have a table of which genes were in the Monty data set. And so doing a table lookup, then we can then take these gene names and then go to the human genome the human genome database and look up these different genes and to see what their function is. And so here they are. Highly significant genes associated with colon cancer. This is a transcription factor that's downregulated by methylation and promotes tumor growth. This gene is a membrane protein, catalyzes silation of glycoproteins. It's overexpressed in colon cancer. Secreted proteins, highly expressed in colon cancer and gastric cancer. And you can go down the list and see what we found here. But that's where we're at with this project right now. This is still an ongoing study. We think that this has the potential to be a very powerful tool. Some questions that still need to be investigated that are active subjects of investigation. With Pepe's help, Pepe Camacho, we want to look at batch effects. Batch effects are things that occur when the genome analysis is done in batches. Different factors, experimental conditions may affect the signal that's measured. So we want to take a look at those kinds of effects. And then we need to then do some studies to see if the genes that we're identifying are significant compared to other studies of gene cancer classification. These are I have a virtual research group that's goes across the globe, and it's been fun and exciting to work with all these folks. And Stuart, as I mentioned, has made big contributions to the code that we use. Heather Teague works at the NIH, and she's our collaborator there. Hamid Abdullahi has been a longtime collaborator with Masa Akbari. Is a postdoc at Nijmegen at Radboud University. She made significant contributions to help our understanding of ASCA permutation. Pepe Camacho has been really helpful with understanding how to do permutations in ASCA with nested designs. So that's it. Thank you all for your attention.
Thank you so much. This is extremely interesting. Let me ask people if there's any questions. I have a number of questions, but let's hear if there's any questions from anyone.
I see
A raised hand. Yeah, Rafa.
Yes. Hello, everybody. Hi, Paul.
Hi,
Thanks a
how are
lot
you?
for fine and you. Nice to see you.
Good to see you too.
I have two questions, actually, Paul. Thanks a lot for your nice presentation. I'm sorry I missed it in Rostock last time, so it was nice to catch it up right now. I have a first question that is a bit more general and regards the fact that you actually use ASCA or ASCA Plus as a tool that I might define parallel to Tucker3 because you use it only for detecting if there is any residual structure in the Tucker residuals, if I understand it well. That means that somehow, anyway, your components in the Tucker model won't be individually associated to the single factors of or the single effect of the individual factors you want to explain. That's probably the main difference you have with ASCA when it is applied by itself in two-way modeling. Have you thought of any way to somehow force, if possible, I'm not sure if it's possible or not, to force the Tucker model to explain somehow to have to be characterized by components that individually can explain single factor effect?
So some kind of constrained Tucker model.
Yes, something like that.
Well, we have done, I didn't talk about that today. We did kind of the opposite thing. We looked at individual triads, and then we did ASCA analysis on the individual triads to see which factors they explained. And so we can see there are some factors some triads which will have a strong model for factor A or B or interaction or both or all. And so if we see a triad that just seems to express variance for factor A, then that might give us some clues. But, there are many, you can't, in an analysis of a variance, you can't examine just one of factor at a time because they occur in combination to contribute to significance. Yeah. And so what we also did was a backwards elimination. I didn't talk about that today. We found, what was that, 160 core values were significant on the Bootstrap. So we would eliminate the smallest one and then do a Tucker analysis or ASCA plus on the remaining 162. And we did backwards elimination until significance was, what's that? Am I getting this right? I think I confused just a little bit, but in a manner you would do backwards elimination in a multiple linear regression.
Yeah, okay. So, framework. Okay, thanks a lot. And then I had just a curiosity regarding you showed the slide that was ASCA plus on nested something. It was I just wanted, because you at a certain point you talked about a random factor that is nested to one of the factor, right? Am I right? It was
Yes.
random factor C. Just to be sure, since you use least squares estimation there for the submatrices, you treat that factor as a fixed factor, right? Am I right on that? It's fixed nested to A.
I believe that's right, but I would probably defer to Pepe on that question.
I think so, because we discussed about that, and he always told me that there is some advantages in doing that. I've never try to investigate that further, but I think that's the way, because as far as I know, if you want to treat a random factor as really random in a framework like this, you need maximum likelihood estimation. You cannot do that by least squares estimation. Pepe tried to explain that to me, but at a certain point I lost him. But okay, that was just a curiosity. Thank you very much. Thanks a lot for the nice presentation, Paul.
Thanks. Can't hear you, Rasmus. You're muted.
Yes. Okay, better this way. So in the Bootstrap data, you had like, I don't know, 28 elements or something like that. Now there's 14,000 genes, which I guess means that most of them are irrelevant.
Yeah.
So how does that, because in the Bootstrap data, you have data which is predominantly relevant. You have a very small fraction of stupid stuff, but here you mainly have stupid stuff. And I guess that affects the clarity of the analysis somehow, right?
Yeah, I think, the risk for finding chance correlations is very high. Yeah. Among the stupid stuff.
But could it be, I mean, so you actually, you selected genes that came out clearly significant. Did you try and then remove clearly insignificant? I'm guessing that a Tucker model on a reduced data set where you have gotten rid of 80% of stupidity could lead to a more significant model somehow, whatever that means.
Yeah, I agree, and I think that's kind of on the list of one of our things to try. Is we've got this powerful. So I would so maybe your comment is maybe getting to the stability of the Tucker model that we've computed. How stable is it relative to perturbations? And I will say that when we do the Bootstrap. When we do the Bootstrap analysis with the Tucker3 model, that's giving us some information about the stability. Because we're scrambling the residuals, without regard to tissue, without regard to subtype and so forth. And the eigenvectors that we're getting out have the same shape. We have some of them are not. And we've excluded those from the Bootstrap samples that we then went along and analyzed. So we look for strong correlation between eigenvectors, and not finding strong correlation, we don't include that Bootstrap sample in the analysis. And it tells you something about the stability to perturbation. We're getting out of 2,000 bootstraps, we're getting maybe 1,500 retained and 500 rejected.
Have you thought about that maybe you need to rotate instead of only doing sign flips?
Yeah.
It's like PCA, you actually do have rotation freedom.
Yeah, so I haven't done any work with rotating these models, and that could be interesting. It completely changes then the interpretation, the shapes of the vectors and the interpretation.
Yes, but I mean, it becomes also in PCA, in my opinion, a little bit arbitrary when you start. You have to make some choices. But so if you rotate it, you could rotate towards the global model, and then you actually don't have any changes. You always to. So anytime you have a solution, you rotation is free, right? It's lossless. So you rotate to maximum similarity to your model, whatever that means, because it's not super well defined, but it could be worth looking into.
Oh, that's a really good suggestion. I like that.
Yeah, I'm not sure it's really good, because I think it's just theoretically it might be correct to do that, but I'm not sure it's going to make a huge difference actually. So whatever.
Well, the other thing too is then if we start imposing either constraints or rotations, what do you do with the Bootstrap confidence intervals? How do you map that?
Yeah, a good question.
Because I think that's one of the real value. The biggest value here, I think, is what we're seeing with the Bootstrap confidence intervals. So
makes perfect sense. Yeah. Rafa, did you raise your hand? I think you.
Yeah. I actually had a comment on that.
Rafa, just one. You don't have to raise your hand. Just shout.
Okay, perfect. So I just have a comment on that, and just a question, Paul. I'm not very now familiar on this, but can you in the Tucker3 models that you construct, can all columns all eigenvectors along each mode, they are orthogonal between them? That is to say, the first column of G is orthogonal to the second and the third, etc. of G.
Yes.
So then regarding that, what you can do is what the people usually do in ASCA. That is to use Procrustes analysis to align the multiple multiple stuff. So you basically do an orthogonal rotation towards the direction that you extract with the original data at each iteration step. So in that case, you are really sure that you are in at a maximum similarity.
I think I don't completely grasp the concept that you're trying to describe.
So basically, because when I use ASCA, what I usually do for determining the statistical significance of the loadings we get, what I use a Bootstrap analysis as well. So I bootstrap, in that case, the original data under constraints. That is to say, I bootstrap within every cell based on the design of experiments. I reconstruct an ASCA model, and then the loadings that I obtain for every factor, I rotate them towards the loadings of the original ASCA model, the one that I obtained with the data. And then I have confidence intervals that are somehow they are taking into account the fact that bootstraps may generate spurious variability.
That sounds really interesting to me. It would take me a little bit of reading and study to understand exactly what you've described, and I would love to learn more.
Okay.
Yeah.
Yeah, I will. I mean, I can send you an email explaining a bit the idea, but that's what I usually do when I use ASCA, in two-way modeling. That's because I want to be sure that's of what Rasmus was commenting, that even if when you generate this variability after bootstrapping, that actually the loadings that you extract are always at the maximum similarity with the one you extract from the original data.
I see.
So your confidence intervals, you might have more confidence in your confidence intervals, because sometimes they might encode variability that you induce by bootstrapping artificially in a certain sense.
Mhm. Yep. I think I have a better grasp of conceptually of what you're describing.
Okay. Yeah.
Right. Any other comments? It makes perfect sense to me that you're using Tucker, but I have to ask if you tried with Parafact.
The Monty paper, the authors of the original Monty paper did in fact use Parafact.
Oh, good for them.
Yeah.
We have a few criticisms of their paper.
Yeah.
They retained 100 Parafact factors.
Okay.
That's for starters. But
yeah. No, but Tucker makes sense. I mean, it really makes perfect sense here because you want to extract all the variance you can, and that's what Tucker does. So it makes perfect sense.
Yeah, so I think, Farnoosh was looking for a sample data set, multi-omics data set that we could use. And, we put in multi-way, multi-omics, and boom, that paper popped up. So they have their code and data is in GitHub. So we were able to, that saved us some work there. So
that's nice. Okay, did you get any comments from them on your work?
Not yet. They're in Korea.
Okay.
And really, their important contribution is this novel data fusion strategy for multi-omics data, which is nice, we think.